First, I would like to thank my colleagues in the organizing committee, especially Dr. Hazza, for his wonderful effort in organizing this meeting and directing an invitation to me to meet our colleagues. Thank you, my colleagues, my friends, and my students in joining us today to speak about one of the very important issues in periodontal regeneration, which is a biologic behavior of human gingival fibroblast in gingival soft tissue and periodontal. First, I'm going to throw light on the cellular changes or cellular challenges, facing periodontal and gingival tissue regeneration, and the feasibility of using gingival fibroblasts in periodontal regeneration as a source of endogenous tissue engineering. And then biologically, what happens if the gingival fibroblast, which is supposed to be isolated using guided tissue membrane, if it's getting access into the periodontal defect area? And lastly, novel experimental and clinical trials, especially our effort in this area in the gingival and periodontal regeneration. Our main two defects or two types of defects with acute cellular deficiency are those needs soft tissue augmentation, gingival recession, keratinized tissue with augmentation, and interdental papillary defects. And those needs periodontal regeneration, which is a critical size periodontal. Now, let's see what are the cellular challenges facing these two. In addition to the most common and the summarized challenges by Kornman in 2000, which is the site characteristics, bacterial contamination, innate wound healing, and the surgical procedure itself, we have a huge area of challenge that limit the regenerative power of the remaining periodontal tissue, which is cellular. Regarding the soft tissue challenge or the gingival defect challenge, we have the gingival tissue in the recession area bounded by avascular and usually contaminated root surface. And this is one of the most important factors that make the gingival recession treatment is very challenging. Regeneration in the gingival defects required challenging vertical extraskeletal bone regeneration. So extraskeletal bone regeneration on an avascular contaminated root surface is very challenging. In addition, it's a non-contained defect. So actually, it's an exposed defect and non-contained, and its regeneration depends on the availability of the soft tissue surrounding that area and whether it's bounded by periodontally affected adjacent sites or not. Challenges, we have the main challenge, which is the regeneration of the three highly specialized connective tissue elements, bone, cementum, and periodontal ligament. At the same time, it's similar to that of the gingival recession. It requires an overgrowth of the tissue or regeneration of the tissue in an extraskeletal manner vertically, and the limited cellularity due to the limited surface area of the remaining periodontal structure. We have only the periodontal ligament 0.1 to 0.3, nearly 0.4 millimeter, which is supposed to provide the wound area with a limited number. Periodontal defect itself is a multi-dimensional defect. The most apical part usually has a defect angulation, which is different from that of the most coronal part. Usually, the most coronal part is the most wide area of the defect, which could be out of or which increase the capability or the osteoconductivity of the cellular element. The excellent, well-controlled clinical cases usually find the most coronal part with a residual pocketing that could be due to the inability of the cells to cross that wide part into the other side in order to secrete collagen to fill that gap with another important issue that makes such defect is very challenging is that the remaining cells on which we rely on the regeneration of the remaining tissue are usually sick cells, are affected by the inflammatory process or the immunoinflammatory process, and still secrete destructive signals even after the removal of the local irritants. That's why we are used to wait for about 4 to 6 weeks following the initial therapy in order to start the surgical procedure. That's because we need that cell to regain its health and start to secrete anabolic mediators to help in regeneration after the surgery. We reported that the periodontal ligament cells continue to overexpress interleukin 6 and matrix metalloproteins 1, 7, 9, and 16 even after removal of the lobe. A huge factor in compromising the periodontal regeneration is the traumatic occlusion. Traumatic occlusion derived from the fact that when the periodontal tissue or when the periodontium is reduced, the normal forces falling on the tooth structure translated by the fibroblast as an abnormal traumatic forces, and this induces the secretion of destructive mediators. This is clear now from the definition of the secondary occlusal trauma. The secondary occlusal trauma is a normal force which is translated by the cells as a traumatic force resulted from a reduced periodontal. All these factors, which is related to the nature of the cellular elements surrounding the periodontium, if you need to compensate that by a cellular therapy, you do have a lot of challenges facing you when you apply the cell culturing into the periodontal defect. The most important of which is a cell protection within an open contaminated periodontal defect, in addition to the complexity of the culture procedure. So we have a very challenging defect. It's very difficult to be regenerated, whether it is a soft tissue healing or periodontal tissue healing. And at the same time, the optimal way of regeneration, which is a cell therapy, has a lot of challenges which limit its clinical use on a daily basis. We need we need to change, we need to simplify the procedure and make it feasible for every dentist in order to get a predictable regeneration. This could be achieved by what's called an endogenous tissue engineering. Endogenous tissue engineering could be applied in periodontal treatment in 3 steps. First, we have to think about or to work on feasibility of using gingival fibroblasts, which is the most vascular, most cellular area, which could be used in an endogenous tissue engineering to treat a periodontal defect. We have to think about the feasibility of using gingival fibroblasts in gingival and periodontal augmentation. And then we have to study the methods of its chemoattraction from the gingival tissue into the periodontal or the soft tissue defects. And then we have to study the clinical application, the outcome of such 1 and 2 procedures. So let's go into the journey of using the gingival fibroblast as a source of endogenous tissue engineering in order to make it simple, practical, and economical procedure. Endogenous tissue engineering is a very important field of regeneration by which we can avoid the ex vivo culturing of an autogenous cells with its sophisticated procedures and the possibility of getting infected and then reculture and so on. And also we can avoid the need for manufacturing a scaffold because the cells in an endogenous tissue engineering manufacture its own extracellular matrix scaffold, and by which we can minimize the invasive clinical procedures of getting autogenous tissue and then manipulate it, and that finally, we can use the body as an own in vivo bioreactor. As you know, all of the cells put in function cannot function except when you apply mechanical forces upon such cells in order to secrete collagen. By using endogenous tissue engineering, we can use the physiological body internal forces in order to stimulate such cells to secrete a lot of biomaterials which could be used to attract cells or stimulate cells for endogenous tissue engineering, like the PRF preparation, endogain, and other chemo. So we can say that we have a paradigm shift here in periodontal tissue regeneration from the competition theory, putting guided tissue membrane in order to isolate cells and giving chance for other cells to grow, into the blood clot stability plus an endogenous tissue therapy. Blood clot stability in order to give a chance for the endogenous therapy to. Now let's see what happened if the gingival fibroblast translocated from the periodontal defect into the defect which we are need to read. Currently, we are used to isolate the gingival tissue according to the Melcher hypothesis. But by our more understanding of the potentials of the gingival tissue, we can use certain favorable elements within the gingival tissue and isolate the other unfavorable elements. So Melcher guided tissue membrane is still in action, but we have to make it selective guided tissue membrane. We don't have to make it totally occlusive membrane. We need to make it to allow the passage of certain favorable elements and to obstruct the other unwanted. The connective tissue of the gingiva have a very high soft tissue regenerative capacity. This is because of the high turnover rate, the huge surface area, the great cellularity and vascularity. So how we can use such elements in order to regenerate the soft tissue. From this area, the researchers at the beginning has a great controversy. Some researchers reported that the gingival tissue lacked the potential for periodontal regeneration, and others said that it has a good role, it could has a good role in contributing to regenerate the tissue. This is the early studies. More it was recognized that the gingiva has the unfavorable elements, which is epithelial cells and gingival connective tissue. It's these two elements, we don't need such elements to regenerate the periodontium. And we have a favorable elements, which is the gingival fibroblasts and undifferentiated mesenchymal cells, endothelial cells, and periosteal cells. By competition, the unfavorable elements prevail with subsequent repair. If you leave that without a guided tissue membrane, the unfavorable elements will prevail leading to long junction epithelial. So if we leave the defect as it is, the epithelial cells will give a root surface epithelialization, and gingival fibroblasts and gingival connective tissue will give is directed the cell synthesis into soft tissue formation, and the defect will be filled with granulation tissue covered with epithelial attachment. We don't need that. So the defect to obstruct the proliferation of epithelial cells and gingival connective tissue, and leaving the gingival fibroblasts and gingival mesenchymal cell to grow into the defect area in order to share positively in periodontal regeneration. So why it is the gingival fibroblast? Why we are intended to allow the gingival fibroblast to fill the defect area? What are the potentials of the gingival fibroblast if you culture it or allow it to grow from the gingival tissue periodontal defect? So, do we have a biologic evidences to start thinking to use the gingival fibroblast in periodontal tissue regeneration? Let's see. But I can say that yes, we have a lot of evidences that drew us into or draw attention into source for periodontal regeneration. Number one, the gingival tissue, the gingival fibroblasts are usually of a neural crest origin. The neural crest cells has an exceptionally wide differentiation capability even after the development of the human being into a various ectomesenchymal cell type. In addition, it could be attracted from its side into the periodontal defect if you put a chemoattractant into the defect site. It could be attracted. It's very close to the defect area. If it's attracted into the periodontal defect site, it has a multi-potent differentiated capability, which is very important. It's a stem cell-like property. So the gingival fibroblast has a stem cell-like property. At the early beginning, we used to say that the differentiated cells cannot secrete collagen. But now we are saying that the differentiated cell has a stem cell characteristics, could be redifferentiated into another phenotype and start to secrete collagen. It has also, like any undifferentiated mesenchymal cell, has an anti-inflammatory capability, immunomodulatory capability, and also contain gingival mesenchymal stem cell within its housing. Gingival fibroblast has also a common biologic potentials with that of the periodontal ligament fibroblast. Both the gingival fibroblast and periodontal ligament fibroblast can synthesize and secrete and break down collagen. It also has the same way of attachment and integration. It reacts with the cytokine the same way like that of the periodontal ligament cells, and also exhibit a dose-dependent migratory capability to the different growth factors of the ligament fibroblast. In an osteogenic media, both the gingival fibroblast and the periodontal ligament fibroblast behave the same way, secreting osteogenic uh, markers, osteopontin and osteonectin and alkaline phosphatase. Both the gingival and periodontal ligament fibroblasts also can express bone morphogenic protein 2 and 4, which demonstrated their capacity of hard tissue formation. So the translocation of the gingival fibroblast into the periodontal ligament could enhance such cells to produce hard. Uh, Surprisingly, the gingival fibroblast has a multifunctional differentiated capability. Uh, it could be differentiated into chondrocytes, chondrogenic media, adipogenic media. It could be differentiated into fat cells and osteoblast in an osteogenic. Gingival fibroblasts could also differentiate into vascular endothelial-like cells and vascular smooth muscle-like cells, which is a very important characteristics because to we need such cells in order to improve the muscular defect. In addition, it has stem cell markers. Here, this study reported that the gingival fibroblast and periodontal ligament fibroblast uh, shows expression of the CD73 and CD79, so other CD markers specific for that of the stem. Now we do have an evidences. Let's see how to use it. What happens if the gingival fibroblast translocated into the gingival periodontal? The main two factors that play with the response of the cells, if it's translocated from one area to the other, is the functional mechanical forces of that area and the specific biological mediators specific for the translocated area. So if the cells translocated from the gingiva, where it's subjected to certain mechanical forces, into the periodontal ligament where it's subjected to other mechanical forces, the behavior will differ. If the cells translocated from the gingiva with certain biologic mediators, its behavior will different if it's translocated into another area, which is a periodontal ligament, with another totally different. So the gingival fibroblast within the gingiva, if it's moved into the periodontal ligament, will be subjected into three factors. Number one, mechanical changes. Number two, biological changes. Number three, the contained gingival mesenchymal cell differentiated into another type of cells, which is either periodontal ligament or cementum or bone, because the ecology is different. Now let's see what are the biomechanical changes which will going to happen if the cell moves to the periodontal ligament. The biomechanical forces and the matrix rigidity. Biomechanical forces of the gingiva is different from that of the periodontal ligament. The matrix rigidity of the gingiva is different from that. This process of the mechanical changes are called the cellular mechanotransduction. Mechanotransduction means that the cells convert the mechanical stimulus into the chemical response. If a mechanical force falling on the cell, it will start to secrete chemical agents in order to function. Uh, Number one, due to the exposure of the periodontal ligament into a higher mechanical forces compared to the gingival tissue, the rate of synthesis of collagen in the periodontal ligament are twice that of the gingival tissue. So if we culture the gingival fibroblast and put it into the gingival tissue, into the periodontal tissue, sorry, it will be exposed to a higher mechanical forces. That's why the amount of collagen expression could be doubled because it's translocated into the periodontal tissue, and the periodontal tissue where the mechanical forces are much higher than that of uh, the cyclic, centrifugal, and shear forces increase the collagen expression of the gingival and periodontal tissue fibroblasts. So it's a force here, mechanical forces, translocated into or translated into a chemical signals of collagen. The tensile force and tensile strain applied on the gingival fibroblasts promote the proliferation of the cells and osteogenesis of both the periodontal ligament and cells. So if we translocate the cell from the gingiva into the periodontal ligament, this could enhance such cells to produce an osteogenic or to induce its osteogenesis. The centrifugal force also stimulates the osteogenic differentiation of the periodontal ligament. Vibration promotes the proliferation and osteogenic differentiation of periodontal ligament cells and uh, gingival fibroblast if it's translocated into the tissue. Now, let's see how does the matrix rigidity sensing by the periodontal tissue could affect the gingival fibroblast. The cells are able to sense the variation in the substrate rigidity. The cell growth in the stiff substrate, like that of the periodontal ligament, exhibit a more spread phenotype. Spread phenotype will allow the cells to secrete much more collagen and upregulate the expression of integrin and increasing the cell function or the more cellular. So the periodontal tissue of a high rigidity will allow the periodontal cells to secrete more extracellular matrix protein, while that of the less rigidity, like that of the gingival tissue, the gingival connective tissue, allow the cells to secrete much less the matrix. So if the gingival fibroblast is exposed to the highly rigid periodontal tissue, it will secrete much more collagen than that of gingival tissue. So transformation of the gingival fibroblast into the periodontal defect could allow for exposure to the periodontal tissue with a more rigid matrix and more collagen expression. So in conclusion, for that part of the mechanotransmission, since the mechanical force and matrix rigidity are totally different for the gingival tissue compared to the periodontal ligament fibroblast environment, using the gingival cells within the periodontium could alter its biologic behavior. Factor, which is a biological changes that could happen if the gingival fibroblast moved into. The main two players in this area is the multipotent capability of the gingival fibroblast, as we said before, and the fibroblast responds to a different biologic mediators that are present within the uh, the periodontal. So if the fibroblast exposed, if the gingival fibroblast went into the periodontium, it would be exposed to the fibroblast related to the periodontium. The most significant or the most important is the periodontal ligament associated protein, the cementum protein 23, and periostine and bone motion proteins, cementum attachment protein, all these growth factors contributing to the formation of the periodontium and could enhance the cells to produce. The third important factor that should be considered is the contained gingival mesenchymal cells within the gingival fibroblast. We have less than 3% of the total population of cells are gingival stem cells with all characteristics of that of the stem cells. Uh, In addition to that, it is easy to be isolated. Its proliferative power is much more faster than that of the bone marrow mesenchymal cells. In addition, it's not tumorigenic. It maintains its normal karyotype even after a lot of passages. It doesn't lose its stemness at the higher passages. So it's a very significant and it's a very powerful cells in periodontal regeneration that are contained within or if it's moved from one area to the other in an endogenous way of therapy. So the gingival fibroblast, the gingival mesenchymal cells, mesenchymal stem cell, it's very important even for the growth of the gingival fibroblast. The gingival fibroblast is very important for the gingival mesenchymal stem cells. This research reported that gingival fibroblasts could act as a stromal feeder for the gingival mesenchymal stem cells. So the presence of the gingival fibroblast is very important for the viability of the gingival mesenchymal stem cells. So if we get a mixed culture, it's much more important than getting a pure gingival mesenchymal stem cell culture. The gingival fibroblast will maintain the gingival mesenchymal stem cells by acting as a stromal cell feeder for that cell in order to maintain. Here we have the very important research of Dr. Karim Fawzi and Dr. Mona Darhous. We are proud of this. The gingival mesenchymal stem cell here used in forcation involvement. Why it's important also to use the gingival and not the pure mesenchymal stem cell. It's a very important point because the gingival fibroblast is very easy to be cultured and to be maintained. Gingival mesenchymal stem cells are very rare. We have only less than 3% of the cells. So we can get a huge number of cells of fibroblasts if we culture it much more than that of the stem cells. In order to culture the stem cells, a very sophisticated procedure needs a massive number of cell multiplication in vitro, which is could results in a mutagenesis of such cells or loss of. In addition, the fibroblast uh, culturing of the gingival fibroblast has a shorter doubling time than that of the mesenchymal stem cells, which reducing the cost and of the uh, production of such cells. So if we get a fibroblast with nearly the same characteristics of that of the gingival mesenchymal cells, with an easier way, it's very important to use such an option, because, as we said before, endogenous tissue engineering will make the procedure much more easier than before. So if the procedure getting much easier, it will be uh, its feasibility of using it on a daily basis much more. Uh, mesenchymal stem cells also needs isolation, which is not the case in case of differentiated cells. Now let's see what are our efforts in this area in order to use such gingival fibroblast. We have three main researches. The first one was in 2008. Without any evidence, they used autologous transplantation of gingival fibroblast like cell and hydroxyapatite graft material to treat an osseous defect. In our recent study, we started in a human being. It's a matter of only publication. We start to use a cultured gingival fibroblast to treat a periodontal defect in a randomized clinical trial, and now we are publishing a paper about in an experimental animal in dogs, studying histologically the use of gingival fibroblast translocation periodontal defect. In this case, clinical parameters were improved 15 months following to 6 years after therapy following the use of gingival fibroblast defect. This is the second work of the this is a current work in publication right now. Uh, so if we need to make the procedure clinically applicable and to avoid the sophisticated culturing procedure in order to culture the gingival fibroblast, which is supposed to provide a significant improvement in the periodontal defect area, if we need to make it an endogenous tissue engineering way, the perforated membrane will allow these elements to be feasible. We can use the perforated membrane in order to allow the gingival fibroblast to move from the gingival tissue into the periodontal defect. In order to make this feasible, we have to use a chemoattractant in order to attract such cells from the gingiva defect area. For soft tissue augmentation, the procedure is already done, headed by Dr. Maguire, who is start working in 2007, ending by this work in 2020, which is a living cell-based regenerative medicine technique for periodontal soft tissue augmentation. So for soft tissue augmentation, it's an old procedure started from 2007, where Maguire used the autogenous or autologous gingival fibroblast in several trials on a scaffold, different scaffold, or without scaffold if he used a cell sheet therapy where he allowed the cells to synthesize its own extracellular. Here in this study, he used the keratinocyte on a acellular scaffold he reported that it's tolerated by the host and can enhance blood vessel formation and cell migration by secreting growth factors. Another study here is used in interdental papillary defect. He used the gingival fibroblast to enhance the papillary overgrowth, which is one of the most challenging. In this experimental study, he used the chitosan-based scaffold culturing gingival fibroblast in order to increase the width of attached gingiva, and he get a very good results here. In this study, in two studies, he used the gingival fibroblast in order to improve the keratinized attached gingiva, and in the same group we used the gingival fibroblast in order to regenerate the tissue around the implant, failed implant or peri-implantitis effect. Now we can conclude that the gingival fibroblast translocation and exposure to the periodontal tissue mediators could allow for its positive contribution in treatment options should be developed in direction of filtering the favorable cellular gingival elements for optimal regeneration. Total isolation of the gingival tissue adopted by the guided tissue membrane protocol should be re-evaluated. Gingival endothelial cells and biologics and its possible role in periodontal regeneration. Thank you so much, my colleagues, and I'm ready to receive any question on my email or on my WhatsApp site or in Facebook site or in the site of the Egyptian Academy of Periodontology. Thank you, Dr. Hala. Thank you all of the organizers that organized this prestigious meeting, and we hope coming year we can meet in person in the El Delta second meeting, inshallah. Thank you so much. Bye-bye.